In this video, we'll talk about really simplified DNA replication. So, if you're looking to learn about Okazaki fragments, you've come to the wrong place. I'm sorry. Alright, so, quick review about DNA structure. Made of nucleotides, each nucleotide carries a base with it, cytosine, guanine, adenine, and thymine. We can number the carbons in the nucleotide 1 through 5, going clockwise from the oxygen. So 1, 2, 3, 4, 5. And DNA is anti-parallel, which means that one strand is pointed in the opposite direction of the other. And the way that we do that notation-wise is we say that one strand goes from 5' to 3'. And that really means that the fifth carbon's on top, and then the third carbon's on the bottom. So 5' to 3', the nucleotides will look like this, with the oxygens pointing up and the phosphate being on the top. On the other strand, it's the 3' carbon that's on the top and the 5' carbon that's on the bottom, but the phosphate's now on the bottom. So we can say that one strand is 5' to 3' and the other strand is 3' to 5'. So in my little picture here, I have 1, 5 to 3, and 1, 3 to 5' strand. And so this is our double-stranded molecule of DNA, and for replication, you need to first unwind the double helix. And there's a protein called DNA helicase which does just that. And by unwinding the double helix, it exposes the bases, it kind of rips them apart and exposes them so that two other proteins, DNA polymerase, can come in and attach to each strand. And here's the really cool bit. Because guanine and cytosine pair only with each other, remember, because they can form 3 hydrogen bonds, and because of the polarities that they have, then the slightly positive and the slightly negatives line up. And thymine and adenine bond, and vice versa, right? And kind of exclusively, the really cool thing is, by using one strand, you can completely recreate the other strand, right? Because guanine is always across from cytosine, and thymine is always across from adenine, if I have one strand of DNA, I can simply put in kind of the missing or complementary bases, and that's exactly what DNA polymerase does. There are free-floating nucleotides around, each with a different base, and what the DNA polymerases do is they basically just attach the complementary nucleotides across from their single strand. So if there's a guanine here, a cytosine nucleotide will come in, and then DNA polymerase will basically just fill in the missing nucleotides as it goes down. And what it's doing is it's perfectly replicating, right, the opposite strand of DNA, which is over here. And the same is true for this strand. And now in real life, it doesn't happen quite this simply. But again, I just want you guys to get the basic gist of this to see why the base pairing is so important. Alright, so again, the DNA polymerase is run down, and they basically just insert the appropriate nucleotides in across from their single strand. But because this is happening on both sides, you end up with 2 molecules of double-stranded DNA when you started off with just 1 molecule of double-stranded DNA. And how I've done this color coding wise is the original strands of DNA were in black, and the kind of added nucleotides to make the opposite strand I have in blue. So we start off with 1 double-stranded molecule of DNA that had G, C, T, A, T, A, T, C, G as it went across. And we ended up with 2 molecules of double-stranded DNA, each half with one of the original strands and half with kind of a newly formed version of the opposite strand. And so in one of our replicated copies, the 5' to 3' strand is from the original, and the 3' to 5' was newly added by DNA polymerase, and the other, the exact opposite, is true. And this is why DNA replication is called semi-conservative. By taking 1 molecule of double-stranded DNA and by producing 2 new molecules of double-stranded DNA, it does so with each new molecule being half made of the actual original DNA. Right? So each of these new copies is half made of the original kind of black nucleotides and half from the new blue nucleotides.